starting with biochemistry. First, it is the chromatin structure. DNA exists in the condensed chromatin form to fit into the nucleus. Fine. DNA loops twice around the histone octamer to form the nucleosome. So, what is nucleosome? It is DNA and histone. This forms a bead on a string appearance. H1 binds to the nucleosome and to the linker DNA, thereby stabilizing the chromatin fiber. DNA has negative charge, which is from the phosphate group, and histone has a large positive charge from the lysine and the arginine. In mitosis, DNA condenses to form the chromosomes. Then DNA and histone synthesis occurs in the S phase. So naturally, when the synthesis occurs in S phase, mitochondria have their own DNA, which is circular and does not utilize the histones, though it resembles the prokaryotic DNA. So this structure is important. DNA having the histone octamer, it forms two loops around the histone octamer, and this is the linker DNA. This is H1. Then the other histone which forms the octamer is H2A, H2B, H3 and H4. This is present twice, so it forms an octamer. Outside the nucleosome core and large and more basic, which is H1. Then we have the heterochromatin. Heterochromatin is condensed and it appears darker. It is sterically inaccessible and transcriptionally inactive. Increased methylation and decreased acetylation. So heterochromatin, highly condensed chromatin is heterochromatin. Bar bodies, inactivated X chromosomes may be visible at the periphery of the nucleus. Where? Periphery of the nucleus. Fine. Then talking about euchromatin, these are less condensed and appear lighter on electron microscopy. These are transcriptionally active and sterically accessible. U andre true, truly transcribed. Euchromatin is expressed. DNA methylation. These are the changes in the expression of the DNA segment without changing the sequence, involved in aging, carcinogenesis, genomic imprinting, and transponable element expression, and X chromosome inactivation. The process is called lionization. DNA is methylated in imprinting, methylation within the gene promoter site, that is the CpG islands. Typically, silences the gene transcription. CpG methylation makes the DNA mute. Methylation makes DNA mute. Dysregulated DNA methylation is seen in which condition? Fragile X syndrome. Histone methylation usually causes reversible transcriptional suppression but can also cause activation depending upon the location of the methyl group. Histone methylation mostly makes the DNA mute. Same. Lysine and the arginine residues of the histone can be methylated. Here what was methylated? CpG islands. Here lysine and the arginine. Histone acetylation, removal of the histone's positive charge. Relaxed DNA coiling increases the transcription. Thyroid hormone synthesis is altered by acetylation of the thyroid hormone receptor. So what happens? Acetylation increases the function. Histone acetylation makes the DNA active. Deacetylation, it is opposite. Acetylation activates, deacetylation deactivates. Removing the acetyl group, this causes the tightening of the DNA coils, decreases the transcription. Histone deacetylation may be responsible for altered gene expression seen in Huntington's disease. Histone deacetylation deactivates the DNA. Okay, there were two diseases: Fragile X syndrome, DNA methylation. Histone deacetylation, Huntington's disease. DNA methylation, methyl group is added to the cytosine, that is a CpG islands, inactivates the transcription. These are the epigenetic changes. Unmethylated promotes the immune response. Okay. Bacterial DNA methylation, where does it occur? At cytosine and the adenine. So methylation protects the bacteria from viruses and phages. Non-methylated DNA destroyed by the endonucleases. Restriction modified system. Okay. Histone acetylation. Acetyl group are added to the lysine, relaxes the chromatin from transcription. Deacetylation reverses the effect. Okay. done with page 1.